Okay, so for me, cognitive neuroscience is really about linking mind and brain. So by mind, it's obviously a bit of a woolly concept, but here I mean things like attention, memory, the stuff of kind of cognition, of cognitive psychology. How is this implemented in the wet stuff of the brain? So obviously these are ancient questions, and I'll give you a brief historical overview. But really the kinds of puzzles that we're interested in here is illustrated by this example from a surgeon called Wilder Penfield, who in the around the 1950s, he was conducting brain surgery on people who were having parts of their brain removed for epilepsy. So epilepsy spreads electrical signals throughout the brain and causes seizures and so on. So literally what they would do is to kind of cut out the focus of the epilepsy. But whilst doing that, partly for experimental purposes and partly to see where the key regions of the brain were, he would stimulate them with little electrical currents, and he would be able to generate these kinds of sensations. So let me...
Developing a surgical approach to the treatment of epilepsy, Penfield began to map the brain and determine which functions of the body were controlled by which brain segment. Among his many discoveries was that he could summon a patient's past experiences by mildly shocking the temporal lobes and stimulating memory.
This is the brain. Now, when we stimulated at 3, you had a tingling in your thumb. You've probably forgotten now.
Not very clear.
Then underneath this, we stimulated, and to my astonishment, you said, I hear music. Then I repeated it without warning you a little while later. You said, I hear that music again. Tell us what you heard.
Well, I heard what sounded like an orchestra playing, and I asked the nurse where it was coming from, and she asked me the name of it. But I said, I know the song, but I can't think of the name of it. And
then I stimulated it again, you remember, and asked you about it, and you hummed it. Will you hum it now? You remember it?
Yes.
Go ahead.
Da 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 da.
Yes, and then when...
So what's happening here is it's stimulating parts of the brain. When he stimulated one part, she felt a twinge in her thumb. In other parts, she starts to hear music. In fact, she say, is there a radio in the room? So if you stimulate your brain electrically, you don't feel, oh, I feel as if my brain's been stimulated electrically in the same way as if you stimulate my fingers electrically, I will feel it in my fingertips. If you stimulate the brain, you don't feel that your head is being stimulated. It feels like you're having thoughts. It feels like you're seeing and thinking and remembering and so on. So the brain has this kind of special status in the sense that, you know, when you don't feel touch on your brain, you feel the world. You feel all your experiences. You hear music and so on. So how does the brain do this? And again, we see clues from this, and the different parts of the brain have different functions, but how are these all pulled together? And that's one of the questions that obviously we're interested in cognitive neuroscience. To some extent, we look at this from different perspectives, like memory, attention, vision, and so on. So here we're trying to provide a brain-based account of cognitive processes. And really, this has been made available in the last few decades by technological advances in studying the brain in vivo. So obviously, for centuries, we've been able to do this in other animals, but in humans, we've had to wait for newer methods and actually safer methods. So Penfield's methods obviously wasn't going to be a mainstream technique. It was also always going to be limited to very particular situations and particular patients. So now we have more lab-based things where we can actually manipulate or record from a living brain. People have been interested in this kind of question for millennia, so wondering where, for instance, our mental, cognitive life exists. So Aristotle thought it was in the heart, so he was aware of the fact that, for instance, that what seemed to be more intelligent animals had bigger brains, but he thought the brain was a kind of coolant system for the heart, in effect, rather than the seat of mental life. Others thought it was in the brain, but obviously people often thought that it wasn't necessarily in the cortex of the brain. So right up until the 18th century, people thought it was actually in the liquid in the brain. And again, this stems back from Aristotle, the idea that there are different humours, so different kinds of liquids that are causing this balance. And in fact, this is a very famous 16th century anatomist who, when he drew a brain, drew it kind of almost like as a cartoon. And it's not that this person was unable to produce this kind of drawing. He can draw the kind of the face and this part in exquisite detail. but he's chosen deliberately not to draw the brain because it was not informative, not meaningful to do so. It would be much better to draw the little holes here, for instance. And even when you go to the early 19th century, the brain looks a little bit more like the intestines. You know, it's, again, this kind of cartoon-based thing rather than anatomy. But people see the brain didn't look like that. There was no point drawing it, because what would you do with that information anyway, I suppose. So how would you actually link together the kind of mind and brain theoretically? Well, obviously one idea is that the two are just separate kinds of things. So again, this is kind of rooted in dualism, and again, kind of certainly Western religion and spirituality, that the mind and the body are separate things that somehow interact. Now, obviously here, studying the brain and the body isn't really going to give you much insight into the cognitive world if the cognitive world is not on this earth, it's somewhere else. So it's a little bit of a non-starter for science. But even at the same time here, at the same time as this idea of dualism was around, people were looking at this in different ways. That basically these are just different kinds of levels of explanation, different ways of accounting for the same thing. So this is often termed dual aspect theory. So here, an analogy is often drawn in physics between light sometimes acting like a wave and sometimes like a particle, depending on how you measure it and how you look at it. So again, how this analogy actually works isn't clear. But the idea is that they're not two different kinds of things. It's almost just two different ways of looking or explaining the same thing. Another approach, which is still popular out there, particularly with some neuroscientists from a more biological rather than psychological persuasion, is reductionism. And this is the idea that actually we will stop talking about things like memory and, for that matter, consciousness and so on when we understand the full biology of the brain. So memory will go, and we'll just talk about chemicals, synapses, reactions, and so on. Again, it's not entirely clear whether that is the case or not. Some people propose this, others don't. But I think certainly we're not at that point now where we do away with those kinds of mind-based concepts. So going into the early 19th century, one attempt to kind of map the brain, if you will, is phrenology. So phrenology wasn't really based in a kind of a scientific method, if you will, but it was based on an assumption that different parts of the brain are doing different things. So the idea is that different parts of the cortex would have specializations, and if you were good or an expert in a certain thing, that part of your brain would grow, and actually the lumps on your skull would grow. But again, the kinds of things that you would have here were not the kinds of cognitive concepts we would have today. So things like love of animals would be one of their modules, as opposed to kind of memory or attention, or the kinds of things that we would be interested in today. And obviously it turned out that there isn't really a relationship between these kinds of things and feeling your skull size. But surprisingly, actually, people who are good at certain things often do have more grey matter in their head. So people who have a very good spatial memory literally have larger hippocampi and so on. So although this has kind of been frowned upon, it wasn't based on evidence. It's been surprising to kind of come full circle with some of these ideas. So phrenology is using, based on this idea of functional specialization. So functional specialization is really the idea that there are different, each part of the brain is specialized for doing a particular task or a particular set of tasks, or for processing a certain set of information. So not all the information it could process, just a subset of that. And really, to some extent, this idea is still contemporary in modern, say, fMRI. I'll come back to critiques of this. So whereas phrenology failed because it wasn't really relying on an experimental method, there were experimental methods that followed, and these kind of have their roots in what was termed neuropsychology, and that term is still there today. Basically, this is studying patients to see how these parts of the brain are specialized. So rather than just getting them or feeling the lumps on the skull, you actually remove part of it by having a stroke, and you see, well, what can that patient not do as a result of doing that? So in the 1860s, Broca reported a patient who lost the ability to speak. He could just produce a single word called tan. That's kind of what he said. So he lost his speech. And Broca concluded that there is, within the brain, a special faculty for speech and for language. He assumed for the whole of language. This is still called Broca's area to this day. What Broca was in effect doing is that he was actually using an experimental method to figure out what the actual functions of the brain are. So this is a complete step change from phrenology, even though it's working on the basic assumption of this one-to-one mapping between function and region in this case. And others came along, such as Wernicke, and said, actually, I found another kind of patient who has problems in speech, but my patient can talk, he just can't understand what's being said to him. So what Wernicke said is that actually there are two language centers in the brain, one involved with speech perception and one involved in speech production, and so on. And this kind of approach is kind of known as cognitive neuropsychology. And what you're trying to do is figure out what are the different kind of cognitive or mental functions within the brain by seeing the way in which you can partition them through damage. There is another key thing about what they were trying to do, which has kind of resonated with some of the modern debates. And essentially what you can do is that you can take two patients, so one who's can't speak but can understand speech, and one who can't understand but can produce. And you can say, well, there are two separate faculties in the brain for speech, one involved in production, one involved perception. But you don't need to know where they are. So you can make assumptions about how the mind and brain works without ever going inside the skull, in effect. You can do that solely from the observation of behaviour. Okay. And this is an important point, because we'll come back to that. And the reason why this is important is that basically a lot of psychology in the 20th century was not really interested in the brain. Clearly people were interested in the brain in biology and so on, but often not in the human brain in other species. Psychology instead went down a different route, and particularly when we talk about cognition, memory, language, perception, and all these kinds of things, what psychology did is that they invented a kind of computer-based metaphor of the mind. So in effect, what psychology was more interested in is kind of studying the mind rather than the brain, and developing these kind of more abstract models of how the mind is constructed. So again, what would happen is that people would think of cognition in terms of a series of different processes. So here, perception, attention, to short-term memory, where attention is kind of a filter of your sensory world to put it into your memory and so on. Or they would develop something a little bit more fancy in which you've got these kind of interactive models where you've got specialization within these nodes, but they're all interactive. Now the key thing about this is that the brain is nowhere in sight here, that you're developing testable theories of the mind without reference to the brain. And in a way, what cognitive neuroscience is trying to do is to pull back a lot of these models and ground them more in theories of neuroscience. And this was made possible by methods that were developed in the last few decades. So in the 1980s, the first functional imaging methods came about. So PET was a forerunner of fMRI, where basically you would inject somebody with radioactivity and you could see how the radioactive tracer passed around the brain. And basically as regions of the brain became more active, you would get more of this tracer in there. That's the basic idea. And also you would have brain stimulation. So PET and fMRI are recording from the brain, whereas TMS is stimulating the brain. And this is a non-invasive method. So non-invasive means that you're not opening up the brain or injecting a person, but it's very much within the Penfield tradition of stimulating the brain and seeing what happens when you do that. Can you create flashes of light, for instance, is one of the things that you do with TMS. fMRI really only developed in the 1990s. So instead of having a radioactive tracer, what you would do is that you can use the oxygen in the blood as the tracer, in effect, or the deoxyhemoglobin in the blood becomes the tracer. So you haven't got to inject something, you're just measuring something that is already there. And I'll cover that in more detail in the next lecture. So these are the kind of methods of cognitive neuroscience that have developed in my lifetime. And basically we can divide them up according to different properties. So the temporal and spatial resolution tells us about how these methods can record or interact with the brain on a time scale, on a fine or slow time scale, and whether or not they happen at kind of the millimeter or the centimeter or the kind of the whole brain level. So here, this is the temporal resolution. So here, these methods, so I'll talk about ERP, event-related potentials, and electrical recording here. So these operate on they have good temporal resolution. So good temporal resolution is being able to detect things at the millisecond level. fMRI has quite bad temporal resolution. So if you're detecting things that are happening, for instance, several seconds, then that's quite slow for cognition. Cognition happens kind of in sub-second time, typically, and fMRI happens between 1 and 10 seconds. So it's sluggish. If we look at spatial resolution here, so some methods, such as lesion methods, are happening kind of at the gross level, so they're damaging whole brain regions, so not necessarily the whole brain, but so like the parietal lobes, the centimetre and multi-centimetre scale. Whereas other methods can happen at the level of individual neurons, and I'll talk about some of those methods in the second part. And invasiveness is whether or not a method is requires any kind of opening up the head, an injection, anything like that. So TMS isn't invasive, even though it's stimulating the brain. Okay? So that's a magnetic stimulation method that's kind of replaced Penfield's approach. So as well as these, you're either recording from the brain or stimulating the brain. They're either invasive or non-invasive, and again, they're either recording the electrical activity of the brain or, for instance, the magnetic property or the blood flow. So that's hemodynamic. And I'll unpack all of these in detail this week and next week. So this all sounds very good. You know, we're kind of interested in the mind, and we're using these contemporary cutting-edge methods. But there's been a little bit of pushback, particularly from the psychologists, that's saying actually this is a bit like the emperor's new clothes, that these methods aren't really going to address the fundamental challenges of linking the mind to the brain. And these are the kinds of critiques that people have expressed. So one is that it's possible to study the mind without studying the brain, or that theories of cognition don't really make any predictions about the brain. Okay, well, we've already seen that that's partly true, that you can study the mind without studying the brain. So is that the end of the debate, or where do we take it? Another critique is that really all brain imaging is doing is just telling us where cognition is happening. It's not telling us how it happens. Okay? It's just like a mapping exercise, but there's no way of reading the map. Right? That would be a kind of a claim. And a related point here is that basically this is a kind of a new phrenology, in effect, and there is a book called The New Phrenology that kind of tries to make that claim. So how would we respond to this kind of criticism? So what people have said is that really that there's an analogy to be drawn between studying the computer software versus the computer hardware. So one, the mind and the brain is like the software and the hardware of the computer. And the argument is, well, you could study the software in isolation without understanding anything at all about the brain, and actually it's something that has been done for 50 years with a relatively high degree of success. And I think, to some extent, that is a reasonable argument, but it's got its limits. And that's that basically the software that you might develop on a computer, there are an infinite number of ways of doing it. Whereas really biology is constraining the way that the software works. There aren't an infinite number of ways of recognizing faces necessarily, or if there are, the way that neurons fire and work is kind of forcing us to adopt a particular solution. So it is giving us potentially those kinds of deep insights. So for instance, one argument that you could have is that recognizing a word, so a written word, can be done in various ways. One is that you can do what you would do in a dictionary, is that you start with the first letter, look up, if it's table, you look up the letter T, then you look up the A, then the B, and so on, in kind of serial search. The other is that you treat all of these different kind of letter units as equivalent pieces of information in time, and that you map them in that particular way. Now, the thing here is that you can do computer algorithms that do both, and they both work, to all intents and purposes, quite well. But the human brain doesn't do both of these. Everybody's brain does one particular way of doing it, and that is the parallel search. So given that there are multiple ways of solving this problem, why is it that everybody's brain in the world has chosen one solution? Okay? And it's very hard to think of an answer to that question without invoking something about the biology of how this works, that's providing constraints in that. And the answer here is that neural processing speed is not operating at a similar level, and actually to do these kind of search are kind of slow, that they invoke other kinds of problems. If your neural processing speed was the same as that of your laptop and so on, then maybe some people would develop alternative solutions for this problem. So although, to some extent, you can study the mind and the brain separately, the brain is actually influencing the way that the mind is shaped, and it's forcing us to do things one way as opposed to another, either because of processing time, because of or other constraints such as that. What about the second argument, that basically all these fancy methods like fMRI are doing is that they're just telling us where cognition is in the brain. It's not really telling us how cognition works. Well, I think that my kind of answer to that, which is partly derived from people like Henson's paper that looks at this, is that if you were to take something like reaction time studies, which is the kind of the standard thing that people would have in cognitive psychology, what you would do is that you would present words on a screen and people make button presses and so on. So if you were to take one of those experiments and say, well, hang on, all this is doing is it's just telling us when we press buttons. It's not telling us how the mind works. It's the same kind of argument, isn't it? And I think that basically what people are getting confused about here is that they're mistaking the data for the theory. So the data here is when you're pressing the button, what's going on in your brain. Your theory is the how. It's the model that you're testing and so on that you'll do. And in fact, it has to kind of logically be the case. So for instance, if you're doing a kind of a cognitive task, you're being shown faces or words on a computer screen, and you're pressing a button. Essentially, multiple things are happening here. Is that your eyes are processing this information, you're generating electrical activity that's emanating from your head, your blood flow is changing, and it's creating changes in your muscle. This is EMG here, and it's then delivering a reaction time. But all these are just different data for the same event. It's not that one type of data, tells you how, and the other type of data doesn't tell you how. They're just different ways of measuring something that is happening. So it would be odd to say that reaction times tell you how things work and not the brain that is generating the reaction time. It's a very strange argument. And in fact, you can kind of illustrate it with this sort of example here. So this is a study of word recognition that was done in fMRI. Basically what's happening here is that you're seeing two words, one after the other, and they're presented very, very briefly. And basically all you have to do is to say whether that's a real word, to say whether this second presentation there is there. So here you say it is. And in between you've just got this kind of visual kind of junk noise just to kind of disrupt things, make it a bit harder to see, if you will. So what the question here is, or what the cognitive question here is are there cognitive representations for representing words that don't care about whether it's upper or lowercase? So in effect, in our mental dictionary, do you have a separate set of uppercase words, a separate set of lowercase words, or is there some kind of stage in which they're kind of pulled together? And the idea is that if there is a common stage, then seeing this word here will make that one faster. That this will, in effect, you will have a faster reaction time for responding to this if it's preceded by this word than if it's preceded by another word, such as table, for instance. Okay? And that will suggest that there is a common code, if you will, for upper and lowercase. What we have at the bottom is the reaction time data for this. And that basically what it shows is that when you've got the same word presented either in the same case or a different case, that basically people are faster. That you're faster at saying that this is radio if you see radio either in lowercase or in uppercase. So that's the difference between the green and the red bar there, is just seeing the word radio twice, okay, irrespective of the case. And basically what you would do is that you would say, oh, here you've got a significant difference, I can measure that with a t-test or ANOVA or whatever it is, and that implies that somewhere within my mind there is a common representation of these two words. Here, in effect, you've got what looks like the same data, but what you've got there is not reaction time, you've just got the activation of the brain in an fMRI experiment. And that is the level of activation. So again, it would be a very odd argument to say that somehow reaction times can tell us how cognition works, but brain imaging can't. Because actually the data just looks the same. And if I were to remove the label on the y-axis, you would draw the same conclusion. It's just different ways of measuring the same thing. But actually you get something different also from it. You do actually get the where information. So you get this region here that's in the visual ventral stream that's specific for recognizing words. And then you can do other things. You can say, well, is this region, for instance, does it care about numbers? So if you've got the word SEVEN, 7, and the digit 7, for instance, would you get the same pattern? And you can do that here, and you can see whether it's happening in the same brain region, as well as having the same behavioral effect on your reaction time. So you could start to use this to understand how the mind is structured. Okay? It's not just telling you where words are recognized in the brain. It's doing something more than that. But that depends on you constructing theories to test about how words might be represented. What about if you turn it upside down? Is it the same region or not? For instance, you can do this. And that's the how, is your testing of this. So what about is it a new phrenology? Well, again, if you read the way that the media picks up on cognitive neuroscience, you actually kind of think, well, it does look a little bit like new phrenology. So here the idea is that you've got that the brain is to blame for teenage mood swings, sex is apparently all in the mind, pain is all in your head, and so on. And this kind of, it's partly trivializing, because actually people have always known that these things were in the brain since the early 19th century, where we realized that nothing was really in our heart or our blood and so on. These are just metaphors. Everything's in the brain. So being able to see these in fMRI scan, it's like, well, who cares? Or knowing where they are. But again, these are kind of almost, well, to some extent, they're phrenological concepts, you know, mood swings and so on, that there would be more kind of intricate things there. There's no one part of the brain called mood, for instance. It's a lot more complex than that. But these kind of portray it in this very simple way. So I think there is a danger of the new phrenology. And basically what's happening here is that people aren't applying more sophisticated kind of cognitive concepts. So rather than looking at, well, yeah, words, when you see a word, it's somewhere in your brain, we can actually make more sophisticated things. It's like, well, how exactly are words in the brain? Is it the same when they're upper lowercase? Is it the same when they're upside down or mirror reflected? You can actually address those kinds of questions. And that isn't, well, you could say it's a very precise form of phrenology, but that is, in effect, a very different kind of question. So what are the kind of challenges, and how are we kind of getting around this? I think there's also been a little bit of a pushback against the idea that different parts of the brain are specialized by the idea that instead what we have are networks within the brain rather than these module-like specializations. And again, this is one of the hottest areas in cognitive neuroscience now, is to be able to analyze the brain as a pattern of connections. So the word connectome is kind of out there, and here you've got things like the Human Connectome Project, where what they're trying to do is to map individual brains. So for instance, there's an equivalent one in the UK called the UK Biobank, where literally they're scanning with MRI 100,000 brains, okay? And they're recording from this people's health history, their cognitive abilities, whether they're intelligent, whether they're moody, these kinds of things, and being able to try and map these in the individual patterns of connections in the brain, but also in the sense of, you know, are some brain regions larger in some people than others, but in a way that you're actually mapping these with very sophisticated techniques and so on. So in a way, this is kind of pushing back from phrenology, is that a lot of these approaches are what are called data-driven, is that you literally present very sophisticated computer algorithms, all the different kind of patterns of connection, and it looks at this and it churns out the answers. It's not the same as having a 19th century scientist kind of think, maybe there's a part of the brain involved in love or something like this. It is doing it in a much more kind of computationally interesting way. And a lot of people think that when this kind of matures, that again will be in a more kind of reductionist situation, that you won't have areas of the brain for faces and so on. You'll have things that are a lot more blurred. So does that mean that there aren't, that specialization in the brain will go when we understand our kind of connector? Some people are kind of going down that route. I don't think that that is the case, and other people who know more than me are kind of saying the same. And basically the reason for that is that the brain isn't fully interconnected with every other part of the brain. So if I take one region of the brain, it won't connect equally to everywhere. It will connect preferentially to some regions and not others. And other people have argued that will force it to become specialized. The only reason in which you won't have specialization in the brain is that if everything connects with everything, and at that point everything can do everything. But the point at which you start to snip away the connections and you've got selective connections between the brains, you will always have some kind of specialization. It probably isn't going to look like phrenology, but nor is it going to look like one massive network where every part of the network does the same.